So now we're in for a special treat. We're going to have a panel discussion. And I think what I'll do is I'll actually ask our panel discussants to come to the stage here. And while they can make their way up and take their seats, I'll provide a couple of words of introduction. First, our moderator is Steve Scherer, Chief of Research at the Hospital for Sick Children. He needs no introduction, of course, in this audience. He's a world leader in human genome analysis with a particular focus on the genetic underpinning of autism spectrum disorder. We are also joined by a couple of discussants. First, Dr. Laura Arbour. She is Professor of Medical Genetics at the University of British Columbia and Affiliate Professor in the Division of Medical Sciences at the University of Victoria. Trained as both pediatrician and clinical geneticist, she's been addressing the genetic determinants of health with Indigenous populations for more than 2 decades. Through her UBC Community Genetics Research Program, she's focused on rare single-gene disorders and complex conditions in partnership with First Nations of BC and Inuit of Nunavut. Professor Bartha Knoppers is the Distinguished James McGill Professor Emerita at McGill University, Canada Research Chair in Law and Medicine, and the founding director of the Centre of Genomics and Policy of the Faculty of Medicine and Health Sciences at McGill University. She's an internationally renowned expert on the legal and ethical issues related to genome research. And of course, they are joined by our 2024 Canada Gairdner International Laureates. So please join me in welcoming them all to the stage.
Okay, thank you very much, Rob. I wanted to first thank the speakers. They really absolutely stellar presentations this morning, and you all stayed on time to give us our half an hour of discussion, so thank you very much. I wanted to begin by thanking and giving our congratulations to the award winners. We haven't heard yet anything about them. So Drs. Balasubramanian, Klenerman, and Mayer have won the Gairdner International Award, and their citation reads as it goes like this: for the fundamental and applied research that led to a revolutionary and affordable method to sequence DNA on a massive scale, which has accelerated discoveries in the life sciences and medicine. So congratulations. Many of us will hear your talks tomorrow and at the award presentations, but everything you heard today that mentioned DNA, RNA sequencing uses the technologies that they essentially discovered about 20 years ago or so. The sequencing technologies based on sequencing by synthesis still predominate probably 70–80% of the overall worldwide market, and I would predict that 10 years out, 20 years out, they're still going to be there. So for all of the scientists who use the technology that you discovered and developed, thank you very much. Now, I do wanted to say one other thing that I think this award is so worthy because it changed the way we think about how we do actually scientific investigation. We thought about doing kind of sequence one gene at a time. We heard about this from Eric Green. But it was really the conceptual advance of having a technology that allowed you to think about parallelizing the sequencing and studying all of the genes or the genome at once. That changed the philosophy of how we understand the genome and conduct our experimentation. So I want to just make that statement. The topic of the session is current and future impact on biomedicine of these technologies. And we're going to invite Dr. Laura Arbour and Bartha Knoppers to come up and give roughly 5-minute presentations, more on the ethical, legal, and social implications side. Laura is a medical geneticist, and we're going to invite her up first.
And I echo, this has been an extraordinary day, and I'm very honored to be here and to be part of this. And I am going to start by addressing 2 of the bold predictions that were discussed this morning by Eric. From my perspective as a clinical geneticist who continues to diagnose patients and also as a researcher who works with Indigenous populations. So that's prediction 6, the application of genomics to all clinical medicine. And then prediction 9, which is benefits for all. And of course, for that, we know that those with the greatest needs are often those who benefit the least from genomic technology. So important for us to be considering that right now. So as a clinician, I took my first faculty position in '97. And that same year, I saw a patient in the nursery with a genetic condition, multisystem. And it took 19 years to be able to finally diagnose him. And it was because of the technology that's being awarded that that was possible. We went on many tangents and spent $5,000 at a time for single gene sequencing. And finally, 19 years later, we were able to carry out whole exome sequencing and find the variant in a disease gene that had only just been discovered 2 years before. So this really drives home not only that patient, but many, many of my other patients, my colleagues' patients, and why we are staying longer than retirement age so that we can go back and be sure that these patients have their opportunities. I also carry out a lot of my clinical works with cardiogenetics. And so all of clinical medicine, immunology, neurology has now taken over. Gene panels are part of their everyday clinical work also. So with cardiogenetics, what I see are just amazing differences in the last 5 to 10 years. But what we see are a lot of panels, and we see a lot of variants of uncertain significance. So we provide also a lot of uncertainty, and some of that uncertainty really is because the variants are unknown. They're from populations, they're from reference data that is really, I should say, it's absent reference data for those populations, which was, of course, touched on with the All of Us study, the importance of that. But for me, I've worked with Indigenous populations for 20 years, and my work has really been about focusing on access to genetic care, genetic diagnosis and care. And sometimes it affects, these genetic conditions affect communities, but most often it's like everywhere else, genetic conditions just occur in families, for example. So it became pretty obvious, and where am I with my time? Already 2.5 minutes gone. It became pretty obvious that this was a priority, and so I was able to be part of the Silent Genomes Project. This is something that a project that was funded through Genome Canada, CIHR, Genome BC, and many others. And this was really about addressing the lack of genomic reference data in the Indigenous population. Wyeth Wasserman, it was his idea, this needed to be done, and he's the bioinformatician who really is continuing to take this probably, I presume, for the next decade. But what we knew was that we couldn't just carry out a reference, we couldn't just sequence without really addressing what the issues were that were important within the Indigenous population. So that meant, first of all, really considering, the rights-based discussion of the United Nations Declaration of the Rights of Indigenous People that actually include a declaration, an article that says that they should have the rights over their own genetic heritage, cultural information. So this is a rights-based discussion. And Indigenous data sovereignty is not something that is new. It is something that is being discussed worldwide, and why that's important. So if we are going to be developing reference data, how do we respect that sometimes we need to step out of the box that we're used to and understand that there should be certain rights and stipulations that are important in this population? So we went through this exercise with a whole lot of discussions with Indigenous people, international advisors, our national organizations, to see how we can make this happen. Our project exposed many policy gaps here in Canada, things that we need to address. What about the individual patient outside of the community who wants to be involved in research or wants to be involved in clinical care? And how do we consider that too? And what we found is with our Silent Genomes project, there were 2 full jurisdictions that could not come into our project. They could not have the opportunity for diagnosis. Their children could not because of some of those gaps. So if we put this all together, what we need to consider that together, The challenges to the benefits to all has to include a village of clinicians, researchers, policymakers, funders, all who can work together to address these issues that will allow Indigenous populations, other populations who hesitate to be part of genomic research or even to partake in genomic clinical care. So we need to think outside the box. We need to think about how can that happen. And I think there's a lot of movement going forward that will allow that to happen. And appreciate the funders who have also understand this.
Thank you so much. That was great. Bartha, you're next. Go ahead.
Morning, everyone. My favorites were also 6 and 9. Didn't talk to Laura. So let's start with the number 6. And it's actually not your usual gloom and doom, bioethics lawyer, negative, be careful, protect, watch out, and all that stuff. Start with a citation or a title of an article that appeared 3 weeks ago. Genetic factors associated with reasons for clinical trial stoppage in Nature Genetics. We found that trials are more likely to stop because of a lack of efficacy in the absence of strong genetic evidence from human populations or genetically modified animal models. A week later, in Nature Reviews Genetics, a call to action, 12 steps to systematically scale up genomic data sharing. Last year, exactly almost a year ago, Government of Canada invests $15 million in first-of-its-kind pan-Canadian genome library. About a month ago, Genome Canada announced a precision medicine health initiative where researchers who would be funded would be asked to ask their participants to allow their data to be shared with this pan-Canadian genome library. So clinical genomics will come alive in an infrastructure sense, like we did with the maps, like we did with the population biobanks and so on, finally in the clinic, but with a database that can be shared so that the next decision is based on quality data. I say Canadian data, but Quebec data, whatever, but you understand that this is a first, that we will start to build a library that will allow us to not only create an anonymized sort of database for quality assurance, but also a protected sort of ClinVar, if you like, for Canadian patients. So we will actually get the patients to say yes, and that way stop waiting for laws that tell us that we're allowed to share sensitive genetic data. So people will speak with their own wishes, and the researchers will return that data to a national library. Finally. So that's a really good story. The number 10 is bothering me, and it was on one of the slides mentioned extending sequencing to newborns. Now we know it's becoming slowly, pediatricians correct me, in the NICU, in the neonatal intensive care unit, that it's increasingly used to diagnose an infant for which there is no diagnosis with consent and so on of parents. But I was in New York last week at the International Congress, their third, you said, I think Eric Green was there as well, on neonatal and on next generation sequencing in newborn screening. And I thought, at first I thought, wow, well, this will really be great for all newborns. But then there's something distinct about newborns. Let's be careful. This is a public health population program. In other words, it receives its mandate from the state to find asymptomatic, at-risk newborns that will receive treatment and need to be treated immediately, usually, at least in the early months of their lives. What is happening is that in all the research projects around the world about can we integrate this into genomic medicine, pediatric genomic medicine, is that they're using, obviously, research consents with the usual 27 pages and with the, we'll give you results back, we'll follow up, we'll provide counseling, all in their research budgets. What happens if you then put next generation sequencing as part of a program that is supposed to identify at-risk asymptomatic infants. People will say, oh, we don't want every baby genetically mapped at birth, and they'll start moving from an implicit consent, very high participation, 97% model of public acceptance that we need to find these children. And then, yes, we get parents, families, everybody to talk, can we treat, should we treat, I'm religiously against this or whatever. Let us think very carefully, because these public health mandated programs with an implicit and implied consent might be adversely affected in terms of what is actually going to be the best interest of all newborns. Remember, this is around the world that they're using this, and often it's only way that you know that a child has been born. In many countries, because the public health nurses have the mandates to go out and test, do the heel prick, they actually know that a child exists. So my slightly negative, and I'm usually very positive, as most people know about everything, genomic and genetic and you name it, is let's think twice about the right of the at-risk asymptomatic child to be found before we even start thinking about next generation sequencing in pediatric endeavors. Thank you.
Okay, I think that question could consume the rest of the panel, but let's move on to our awardees and invite them to each give a 5-minute overview of what they found interesting in the session, or a question they want to ask, an experiment they want to do, that kind of thing, a future vision, tell us where the field is going, whatever you please, but in just a few minutes, please. Why don't you go first? Yeah. We'll just go down the row.
Right, okay. Well, I mean, firstly, just want to say what a brilliant set of lectures this morning, actually. It was really inspiring. And to remember, as sort of someone involved with helping create a technology, that's only a part of the story. It's knowing what to do with it that really brings impact to the world, and that's what we heard about this morning superbly. So my reflections on this morning is, I mean, one thing was apparent from all the lectures, and that is we're now doing things at scale without even saying we're doing it at scale. It's taken for granted that we're doing things on a population scale, whether it's a population of cells, one at a time, or a population of people. So it's remarkable. 20 years ago, it would have been hard to imagine that you routinely hear lectures on doing things at scale. I think what's apparent in terms of the direction of travel, technologically and also in terms of measurement, was put very nicely in a couple of talks, and I have to declare an interest in epigenetics that I have, and it's really getting more information from cells at scale and more information, I think, from clinical samples at scale. We heard some very nice talks this morning about genetic information from DNA, epigenetic information, which you can also get from either DNA or from histones, as we heard. RNA, of course, is sort of giving us more and more discoveries every year in terms of its function. There's the 3D structure of the genome, which Sarah alluded to from High C. So I think one direction is to actually get all of this information, but we need to get information in a way that's correlated at the cellular level or at the fragment level, because that provides us with mechanism and dynamic information about biology and causation. So I think that's a direction of travel, both technologically and in terms of application. I think the other thing, thinking about the future, some nice talks that told us about linking population scale omic information with healthcare records. And I'm most familiar with the effort in the UK in Genomics England, which was I think, one of the early projects of its kind. This is part of the NHS. Unlike the US, we have one system, so it actually enables this type of experiment with fewer barriers. And we have the UK Biobank, which is 500,000 patient samples that have been whole genome sequenced, and many other things have been measured. Where you have patient records, you need to wait for a generation or two to really see the outcomes of the experiment. So I think part of what we're going to see in the future is, with all of these projects around the world, where we're integrating data, but it's still a moving picture. We still haven't seen the outcomes, people. So I think a generation or so from now, we're going to learn so much more from these studies.
Okay, thank you very much. Dr. Klenerman?
So I think it's just fantastic to see how the field's evolved. So literally we wrote this idea down on a piece of paper in 1997. And so to come to a meeting like this and see how it's been used by people all over the world is very gratifying. And we've obviously reached this point where we can start to use it in medicine, which is very exciting, but also creates, I think, some ethical issues that we need to think through. So it's great if we can sequence people and work out what's wrong with them if we have a treatment for them. But we haven't really discussed if we sequence them and discover what disease they've got, and there's no treatment for them. So we work on Alzheimer's disease, of which there's just starting to be some therapies which aren't very effective. So the question is, would you want to know whenever you're sequenced, newborn or 18 or whenever, that you're going to develop Alzheimer's disease if there's no way of curing that disease? And you might want to know because you can put measures in place before you lose your memory, so your family could live better. And you might not want to know that at all, because I think in some instances people just stop living when they've been diagnosed. So I think that's a serious issue for us as we start to use the technology more widely. And I think the other issue was people alluded to lifestyle changes if you knew you had a disease. And I think the big problem in the UK is if you're wealthy, it's very easy to have lifestyle changes and exercise and eat less, but it's the people who, in the poorer parts of our community, who can't change their lifestyle. And so, if we're going to start using polygenic risk score to predict people if they're going to develop disease, we need to put measures in place as a society to ensure they can make the lifestyle changes they need to.
Okay, thank you very much. Before we move to the next awardee, quick comment, because I was dying to ask both or all of you, when was the eureka moment? Was it 1997 when you first conceptualized this? And
I think there's a slight difference in the eureka moments, because it was a sort of combined effort. For me, it was in August 1997.
Yeah. Well,
this is a Discovery Award, I think.
So.
Yeah, my answer would be different. I think the eureka moment is a bit mythical because ideas evolve over time, and there's a lot of iterative trial and error in the process. So most eureka moments are actually years in the making.
Okay. And just to point out, of course, Peter St. George Hislop, the expert on early onset Alzheimer's, in the audience, so he may have answers to some of the questions that were raised. To the next awardee, Dr. Mayer.
Thank you. So I found also the talks very fascinating and inspiring, and it many details that are fascinating per se, but also one thing that emerged, in my opinion, on all the work that is done by everybody here, is that the more we dig into biology, the more we discover complexity. And I remember when we, back in 1999, 2000, and so when we went to see the most prominent geneticists by telling them, guys, we are going to solve your problem. We are going to be able to sequence a full genome within a day for $1,000 because we wanted to make some benefit, cost it less normally. They literally raised their hands and saying, this will be of no use. We won't be able to do anything with the data. It's too much. We don't know how to treat them, which didn't help to promote to convince our investors and backers that it was worth doing this, because the geneticists wouldn't know what to do with it. But today, I think it's my turn to raise the arms and say, what will we do with all that knowledge that you are generating, all that complexity? And actually, but I know the answer. I know that there will be some sort of biome GPT that will get all this data together, and that we will interrogate what is, design me a drug for that disease or for that condition. And it will do. I mean, I guess, like myself, all of us have tried one way or another to use ChatGPT for one reason or for another. And I tried a couple of times when I was lazy going into documentation for programming. I said, write me a piece of code for making that little part of program. And he said, yes, of course. And he gave me code which was total fantasy. Mm-hmm. And I think that's what may happen in the end with the Biome GPT. It will always give you an answer, but you will have to test it experimentally in order to validate it. There will be no way of being sure that the answer is correct. And this will bring us back to, I think, a stage where we need to go back and make really phenomenological experiments and try not, we will be able to explain everything, but I'm afraid we will not be able to predict anything out of all that knowledge, which is, of course, important to have, but there will be more needed, and what will be needed is experimentation.
Right, and just to remind you, many of us in the audience thought your technology was a total fantasy too in the early days, so perhaps fantasy is just complexity, and I just don't know it at the time. But your points are very well taken. How much time do we have? Do we have a couple, 5 minutes? Okay, we're not gonna, we have lots of questions that have come in. I want to come back, because it was raised here by at least two of you, the issue of perhaps doing a newborn screening program and identifying genetic variants for which there's no treatment, even for which there may be a treatment, and the treatment may not be accessible to all those people who are involved in the screening program. My question is to Laura and Bartha. Do you want to just comment on that? Is the time really right? You were at the meeting in New York last week. What's happening worldwide? Because I think this could be the game changer for uptake of this technology or suppression of this technology. You can use a microphone.
Well, it'll definitely take away from the universality success as defined by WHO of the biggest, most successful public health, international public health initiative, if that were to be the case. So I'll skip straight to what is my guess, what that we could do for now to save the good thing that we've built, which helps all asymptomatic at-risk children to be found, not touch that part, and preserve and pay for and protect public health programs for classical, what we could call tier 1. I don't like the word tier because it's always leveling, but classical, and then decide on a consent basis rather than making sure the newborns all get tested irrespective of screened, irrespective of parents, then bring in the extra sequencing and everything else that we think will be of help or may be of help in pediatrics where the parents and the families and so on are involved. That brings it to the clinical, whereas I'm worried about the public health programmatic aspects of it. So maybe if we split it off to sort of the safe, tried and true, that's a benefit to all children, and then where you can afford it and it makes sense and you can actually explain it and there's something you can do about it, leave it to treatable during childhood for now and offer new technologies as a healthcare system thinks it's a priority or not.
Okay, Laura, and maybe you also want to comment on the need for a Genetic Non-Discrimination Act in some countries. For example, we have here.
Right. So that's an excellent point. First of all, I just want to say I absolutely agree with everything that Bartha has said, because where I see the challenge is in the second phase of it. If we do decide to go with whole genome sequencing for newborns, I'm just wondering, of course, in our own healthcare system, whether that could work. And I'm thinking that what we are going to see is very much a two-tiered way of dealing with it. The Genetic Non-Discrimination Act, I'm glad you brought that up. This is an important act in Canada. We thought by now it would have been challenged. I mean, it has been challenged, but it is still in existence. And what that says is that in certain areas, that employers or schools, for example, no one has the right to obtain your genetic information and cannot ask for it. You might want to comment more on that.
Okay, we have one more minute. So as the chair, I get the last question. Sorry. So I mean, your technology that you developed, and it's evolved over the years, but it's the same chemistry, actually, I think, in different boxes, right? Scaled up in different ways. What's your next dream technology or fantasy technology? Back in the day when I was a graduate student, I thought maybe we could invent a technology that could read the sequence directly off the chromosome. That would give you the best accuracy, right? Because there's no chemistry involved. But do you want to just share your thoughts? I know there's a lot of students in the audience here, but share your thoughts on that. What's coming and maybe how we're going to get to the $100 genome that's 100% accurate.
So I'll make a case for the 6-base genome.
Okay.
And I'll declare an interest here.
Any stock tips? No.
Something I helped create, and it's actually now a product in a company called Biomodal. So this is a way you can use any sequencing platform, including next-generation sequencing, and it gives you, with high accuracy, all the genetic bases and variants, methylation and hydroxymethylation. So I see a future, actually, this is sort of part of the vision towards measuring more than one dimensionality of biological information simultaneously on the same molecules at the same time. So I think, this is just being put in use now, so I think this is going to give us different information.
And just for the people who don't think a lot about this in the audience, the methylation differences are a way to sense environmental changes and influences. So, next.
We focus much more on proteins than being able to, I mean, one would like to be able to sequence proteins in a very straightforward manner, but we put a lot of effort into being able to identify very rare proteins or protein aggregates, particularly associated with Alzheimer's disease. So I think the DNA sequencing area is probably maturing, and there are other areas such as proteins where there's a need for much more technology development to detect rare proteins in biofluids for early diagnosis and this type of thing. So I think certainly that's where we're putting our effort, and I think that's where maybe the future is non-DNA, I think.
Great. Excellent prediction. And last?
I must say I have no answer to that question. And that's why I need also to declare an interest. What I'm doing presently in my company is that it is to skip a little bit the genetic and genomics and whatever, omics knowledge, and to jump to hypothesis-free treatment development. Because, that's my iconoclastic way of doing things.
Okay, and then to close, I just have a yes or no question for each of our award winners. Whenever it was 20 or 30 years ago today, when you were grinding away in the laboratory, did you think you'd be sitting on the stage here, winning such an important international award? You can just say yes or no, or if you want to annotate, go ahead. I don't think Janet will pull you off the stage.
Simple answer, no, that wasn't really any part of what I was thinking at the time. We were just trying to figure out how to make it work.
Just try and do some good science, yeah? Next?
I would say the same answer, no. We had no expectations that it would take us this way.
And?
Well, on this, to be honest, when we got the first results and realizing that if we got it to work, which was not initially the case, it took time, but knowing from the basic experience what the potential was, I was wondering if I would not end up in such a stage, I must confess. It's when you see that taking the numbers, making the calculation, you see that you go from 1 to 5 basis per second to million basis per second, you see it should have an impact. I hope it's not taken for lack of modesty, it's just what came out from the calculation is that we would make a very big jump, and I would be lying if I would say it. I wouldn't think it would make an impact, and that at some stage, at some point, it would be recognized.
Maybe that's encoded in your DNA. I think all of us see all three of you as visionaries in the field of biology as a whole, and congratulations again on the award. Thank you to everyone.